In advanced chronic kidney disease, a protein-restricted diet is recommended. The effects of such a diet may be enhanced by keto analogues. But why is a protein-restricted diet needed in chronic kidney disease, and how do keto analogues work in this context? Proteins are fundamental components of the human body. About 15 to 20% of our body is made up of proteins. For example, proteins in our muscles are responsible for contraction of muscles and thus all our movements. Our immune system uses antibodies for defense against viruses and bacteria. Antibodies are large Y-shaped proteins. All enzymes which we need, for example for digestion of food, are proteins. In summary, proteins are the main machines in the human body that do most of the work our body needs for life. But how does our body produce all the proteins it needs? Proteins may form complex three-dimensional structures, but basically proteins are formed by strings, like a ball of wool is formed from yarn. The strings are formed of smaller units like a necklace formed of pearls. The pearls are amino acids. Amino acids are small molecules with an amino and an acid part. To build proteins, our body uses amino acids from food. After a meal containing proteins, these proteins are broken down to amino acids in our gut. The amino acids are used to form new proteins. Usually, the composition of amino acids in our food does not perfectly match our body's needs to make proteins. To compensate for this, our body can modify some amino acids and rebuild the ones it needs. Not all amino acids can be produced in this way. Essential amino acids have to be taken up with our food. The liver can rebuild some digested amino acids according to our body's needs. In a process called transamination, the amino group of an amino acid is transferred to a precursor in order to form another amino acid. This precursor without amino group is called keto analog of the respective amino acid. Keto analogues from the metabolism cannot be used to build essential amino acids in the liver by transamination. The newly formed amino acids can now be used to build new proteins. The proteins of our body are continuously recycled. Amino acids that are not needed anymore and cannot be recycled are broken down to produce energy. Thus, we need to supply a certain amount of amino acids daily with our food. During the degradation of amino acids, the amino group is cut off. The released amino groups form ammonia, a toxic molecule that is immediately rendered harmless by converting it to urea. Urea is excreted via the kidneys. In chronic kidney disease, the kidneys cannot excrete sufficient amounts of urea. Thus, urea accumulates in the blood, leading to uremia, which causes many symptoms of chronic kidney disease. A low-protein diet reduces amino acid intake. This decreases the amount of excess amino acids and urea waste. Still, a minimum supply of amino acids is required to replace aged proteins during normal turnover. Thus, urea is still produced and may accumulate. To further reduce urea production, keto analogues, the precursors of essential amino acids, can be supplied. Keto analogues basically are amino acids without nitrogen. They can be ingested as tablets with regular meals. Keto analogues are taken up in the gut like regular amino acids. Then they can be converted into amino acids according to the body's needs. Keto analogues of essential amino acids can be converted to essential amino acids, reducing the need for protein intake. This is achieved by transamination. The nitrogen-containing amino group of a waste amino acid is transferred to a keto analog to form a new amino acid instead of being converted to urea. As a result, urea production further decreases and uremia is ameliorated. Keto analogues only work in combination with reduced protein supply. Only then our body is forced to make use of keto analogues and nitrogen wastes to form amino acids. Clinical studies have shown that a protein-restricted diet plus keto analogues delays the need for dialysis treatment. Furthermore, it ameliorates uremia, metabolic acidosis, and disturbances of the calcium phosphate metabolism.